Good afternoon. This afternoon's theme continues many of the themes that we discussed this morning, albeit looked at from different angles. Just by way of introduction, I wanted to say the following and present or pose the following questions. Could we have the slide, please? So much of our thinking has been focused over the last decades by this scheme of Bert Vogelstein and Ken Kinsler, in which one views the histopathological progression of a carcinoma in this, or a tumor in general, in this case the context of the carcinoma, as yielding a primary tumor, with the question being of what are the final steps that enable cells from a primary carcinoma to metastasize. And subsequently, as first enunciated by Josh Fidler, one described the invasion metastasis cascade that we've alluded to on many occasions today. Here's yet another depiction of it. It's an extraordinarily complex sequence of events, and indeed the complexity of the invasion metastasis cascade rivals the complexity of the steps that preceded it and led to the formation of the primary tumor. And that, of course, raises the question of how cancer cells are clever enough to acquire the multiple attributes that are required in concert in order to successfully execute all of these steps. In fact, one might begin to partition the invasion metastasis cascade into two major steps. One involving the physical translocation of cells from the primary tumor through these multiple sites and channels to the site of initiation of formation of a micrometastasis. And independent of that, the ability of a micrometastatic cell to effect the process of colonization and thereby generate a macroscopic metastasis. And an important solution to these problems has come from invoking the epithelial-mesenchymal transition, the EMT, which is resurrected opportunistically from early embryonic genes, because, in fact, there's every reason to believe that the epithelial-mesenchymal transition program enables a carcinoma cell to execute all of these steps. This has not been proven, save for the last one. And if that's true, that represents an enormous conceptual simplification of how the last steps of malignant progression proceed. The last step involves, by necessity, the ability of a cell from one target organ, or one primary tumor organ, in this case, let's say the breast, to adapt to a foreign tissue microenvironment, such as that of the bone marrow or the brain or the liver, a topic that at the end of today Jean Massaguet will deal with. Still, having embraced the EMT as an attractive solution to the problem of how these multiple steps are executed and how cancer cells acquire the ability to do so, this leaves a number of important questions outstanding, and I thought I would just list some of them, not with the pretense that they'll be answered today, but that they will be agenda items for this field of research over the next years. And that is, are the genetic changes that occur within a primary tumor sufficient already to enable it to physically disseminate, or are additional genetic changes required? Indeed, there's every reason to believe that once one forms a primary tumor, that those cells may already be able to undergo an EMT. Does the EMT program represent a good explanation for describing the acquisition of malignant traits by all types of carcinoma cells, or is it an idiosyncrasy of only a subset of them? Are there alternative programs which are equally important and influential? We know that in many cancer cells, the EMT program is induced by heterotypic signals that originate in the stroma of a tumor. Here they're called contextual signals, yet their nature is poorly defined. Certainly TGF-beta has been invoked there, but TGF-beta is only one of a number of signals that seem to conspire and work in aggregate to achieve that effect. Another critical question is, what are the changes that must occur in the cancer cell during its evolution as a primary tumor cell in order to make that cell responsive to the EMT-inducing signals? We still understand very little about that. And does the EMT program, as I've just described in a very hypothetical tone, enable a cancer cell to execute all of the steps of the invasion metastasis cascade except for the last one, or is it really more complicated than that? And that also is a question that is speculated on but hardly proven in any direct way. How do non-carcinomatous tumors acquire the traits of high-grade malignancy? As attractive as the EMT program is in describing how carcinoma cells might in principle acquire highly malignant traits, this doesn't really deal effectively with how non-carcinomatous tumors acquire these traits, and that was dealt with in part this morning when, for example, we heard about from Sean Morrison the progression, and from Eric Sahai, the progression of melanoma cells. And therefore, it's unclear how universally applicable the EMT program is. Is it true that carcinoma cells that undergo an EMT acquire stem-like attributes, or is that only an idiosyncratic response of a small subset of carcinoma cells? A very knotty problem is the following, and that is when a cancer cell disseminates from one tissue site to another organ site, it must acquire a number, or must develop a whole number of adaptations. And given the number of diverse primary tumors that can in principle metastasize, and the number of sites to which they might disseminate and eventually adapt, how can one begin to rationalize this? Are there certain universal changes in cancer cells that enable them to disseminate and adapt to all tissue microenvironments? Or are there different pairs of such combinations and therefore enormous complexity? How do cancer cells, once they're disseminated to a foreign microenvironment, how do they learn to adapt to what is initially for them an incompatible microenvironment? Do they need to undergo initial genetic mutations, or are epigenetic changes sufficient to enable that to happen? And is it the case that there are certain cells that leave a primary tumor, and from the moment they have left the primary tumor, are they already adapted to grow aggressively in a distant site, or must they also undergo a series of evolutionary changes in order to enable them to adapt subsequently and with great delay to the foreign sites to which they've disseminated? Some of these topics, some of these issues will be touched in passing today by our speakers, and others will represent fodder for research over the next years. Hopefully by the next time we have one of these invasion metastasis symposia, some of the answers will be presented.